Greetings, everyone. It's Mr. Gopal here, and I want to talk today about genetic mutations. So we've talked about vaccination, we've talked about inheritance, we've talked a little bit about mutation, but there's more to say about this topic. And just like the virus, we've talked about how coronavirus COVID-19 has mutated, and as a result, we've had to find new ways to treat it or attack it. And maybe the initial vaccine, it's the jury is still out on how effective it is on the variations, the mutations of COVID-19. Just like a virus can mutate, a human being's genetic material can mutate. And that takes on several forms and has several implications. And that's what we're going to talk about today. So this is part of Unit 6 on Genetics and Biotechnology. We want to answer the question, what are mutations? What are some of the different types of mutations that occur in genes? How can we use mutations in DNA to compare genomes of different species and then infer how closely or distantly related they are? So it might surprise you that I think our DNA and the DNA of bonobos monkeys is like, I want to say it's over 99% similar. So the variation that makes humans humans and bonobos monkeys, it's less than 1%. But let's consider the scale here. From time to time, when we're talking about genetic material, we could be talking about 2 million base pairs of nucleotides. And in another video, I will get into the different divisions in which we examine DNA. Nucleotides is one of the most micro levels. All right, we can break base pairs down even further based on the different elements involved, you could say. But we build up and we get genes, we get chromosomes, we get the entire double helix, which could be very, very many million base pairs in certain very specific and implicative series and orders. Okay, so first of all, to step back from that, that'll be covered at a different time. We want to look at mutations, and so I also want to use this video as an opportunity to show you how I annotate for important material in a text. All right, so much of studying living environment and biology is text study. All right, let's go over how to break down this text and use it to answer some crucial questions. So they give you the example. I love these CK12 assignments because they give you an example of something very specific, something relatable. I would say maybe thinking about a hairless rat is not necessarily relatable to us. We see some very hairy rats on the subway tracks, I'm sure. But what's interesting about it is it gives us a way in, something immediately kind of eye-catching and head-turning to think about the idea of genetic mutation. All right, this rat is hairless because of a mutation to its DNA. We're going to talk about much more relevant mutations to human beings and their implications. Okay, so they break this down, and as we break this down, I want to talk about what it is that we're looking at in order to understand that a mutation has occurred. Okay, so what might surprise you here is the fact that we can divide mutations into categories, and by far the most abundant category, I'm just going to underline it, is what we call the neutral mutation. All right, so I'm going to underline that. Actually, let me undo that. I'm going to box my vocabulary. All right, so important vocab word here, I box it. I go back. One of the advantages of studying during these times is that you can make it zoom, you can annotate, and you can even just screenshot your annotations, which is very helpful, I find. The majority of mutations have neither negative nor positive effects on the organism. All right, and so that is our definition that goes along with neutral mutations. All right, so it might surprise you to know that. All right, that's our first type of mutation. And many other mutations, they say, have no effect on the organism because they are repaired before protein synthesis occurs. I'm going to circle this one, or I'm going to box this term as well in red, protein synthesis, because we already talked about this And so we talked about it in a different lesson. It'll come up again. It's very important in biology. I'm just going to make a little arrow above and remind ourselves that protein synthesis is the process of DNA replication. So DNA replication that occurs in cells. All right, and so probably you remember a few aspects of this process. DNA is outright copied, but then there's sort of this turnkey process for different cells in the body that you may have looked at that unlocks only parts of the DNA sequence because these cells serve different purposes. So if I'm talking about a blood cell, when those are replicated, the DNA is replicated and created. We have the same genetic material being passed around for all types of cells across the body, but something that we need to understand is that a blood cell has different purposes and therefore needs different sections or regions of this DNA sequence. So only parts of it are unlocked, and those parts are relevant to it being a blood cell. And that might be different from, say, a cell in my saliva. Even though they're both liquid, they serve very different purposes. So when we talk about protein synthesis, we're talking about protein DNA replication and creation. So we replicate it, we pass it on to other cells. Parts of it are unlocked based on the purpose of that cell. And I hope that's clear. Going along, if a cell's DNA is permanently damaged and can't be repaired, the cell is likely to be prevented from dividing. There it is. Another beautiful aspect of the body is that there is a built-in mechanism to check on the health of a cell, and it will be prevented. Of course, as we go on to certain harmful mutations, we're going to see that this process doesn't always work. So I'm going to clear these annotations, and I hope you're getting something out of this process. I think before I clear them, I'm just going to point out one other thing, which is that I try to be consistent in the way that I annotate. I try to box vocabulary. I try to underline important things. In the margins, I'll put question marks over parts that I feel like I don't understand and I need to study again. Or I might even just kind of write things in my own words. So use the white space around paragraphs in order to remind myself of things, ask questions, make connections, things like that. So I'm going to clear this. And we can see this repair process taking place. So I'm looking from top to bottom here. We have a damaged part of the nucleotide. That part is cut out. And the surrounding, what's hard to understand is that the surrounding proteins are cut out as well and then replaced. And they add normal nucleotides. So even though there were just these two pairs or two nucleotides that were damaged, they cut out the surrounding, let me see, surrounding 2 on each side of it as well. This is a more effective way to ensure that the DNA strand is not consistently damaged after the repair is done. So they kind of overcompensate. And that's how I interpret that picture. So quickly, I want to talk about the difference between beneficial and harmful mutations. So beneficial mutations are absolutely crucial for evolution. And so here are the rough points that you want to be aware of. I'm going to underline the relevant parts of the passage, all right, and then just talk about what they mean. So actually first, let me continue my tradition. I'm going to box vocabulary. I know that this is important because they bolded it, beneficial mutations, and then my definition of beneficial mutations, that's mutations that have a positive effect on the organism. Okay, and notice that I'm not underlining the entire sentence. I feel like I'm getting the gist just from that part. The reason they're essential, we need genetic variation in order for evolution to occur and for there to be the standard level of competition that results in more adapted individuals persevering, okay, or winning out, you could say. So let's go over that process. Two different, I guess, like two genetically varying members of a species are competing within a certain habitat. And the one that is more resistant, let's say, to a certain disease because of their DNA variation is the one that's going to survive. And that is good because future generations will inherit that DNA and be more resistant to a certain disease. I'll point out another example. There was the black pepper moth, which you probably saw from your study of evolution earlier in the course. The idea with the black pepper moth is that this is a moth that adapted to the smoggy conditions of the Industrial Revolution and evolved to camouflage itself. And so it turned darker and darker until it could blend in on tree bark with its, I guess you could say, on blackened tree bark from the smog. And it protected itself from other animals by being disguised. It was essentially invisible. And moths that did not evolve to darken themselves in accordance with the change in the atmosphere, those are the ones that perished. What we didn't know before is that mutations in the DNA are responsible for these changes in the black pepper moth and anything else that is evolving. All right. And so they give a few examples down here. I'm just going to underline things that I find important as well. I'm going to use the drawing tool for that, and I'm going to use green. So we know they're essential for evolution. They increase organisms' chances of surviving or reproducing. All right, and then they give two examples down here. So I'm just going to write that line and then just say examples EX. All right, that is the way that I would annotate. Again, I would just screenshot this stuff and move on. And now we look down at harmful ones, all right? So the analogy they use, I like it. Harmful mutations occur the same way that maybe a random change to the engine of a car, which is very complex, would occur. If we make a random change, the odds are maybe it might benefit, maybe it would do nothing at all, but there's a substantial chance that it might actually damage the car, okay? And this is what happens sometimes as well. Sometimes there's a genetic mutation response to conditions that allows us to persevere or improve our ability to adapt and survive. Other times the change is random and the change is harmful, unfortunately. So this is one of those times. And so we see here, they don't write the word, so I'm not going to be able to box it this time, but I am going to underline what I think is the most essential part. Any random change in a gene's DNA is likely to result in a protein that does not function normally or may not function at all, okay? And now I'm going to close with just explaining why I underlined that part. And what I'm going to write on the side, I'm just going to put an arrow and say that that is my definition of harmful mutation. Sorry that that's partially, let me move that a little bit. There we go. And the reason I underline that is because that gives us the gist of what's important. How do I know that's the part that's important? Because it tells us the consequence of a harmful mutation. And then the final thing I want to do in this video is just discuss the difference between what we would call a genetic disorder and something like cancer that could be like a predisposition to cancer could be inherited, but it's not a genetic disorder. So I'm going to clear this so I can scroll down. And now, they say a genetic disorder is a disease caused by a mutation in one or few genes. You probably guessed what I'm going to do. I'm going to do the same thing that I've been doing. I'm going to box it in red. I'm going to underline the definition so I have it for my notes. Genetic disorder, disease caused by mutation in one or a few genes. So I think that this part is especially important. So I'm going to make an elaborative note here. And we're just going to say that that's very few genes involved. All right, that's one thing that I want to remember about a genetic disorder. On the other hand, they describe cancer, which we've studied before. And on a basic level, cancer is caused by a malfunctioning cell. All right, there's no kind of termination impetus in the cell. So normally there is a process by which cells are regulated and they don't just kind of continue to multiply willy-nilly. Cancer kind of removes that self-regulation. And so it says because of the mutations, and those are genetic mutations, cells with damaged DNA are allowed to divide without limits, and cancer genes can be inherited. However, cancer is a little bit different because it's not out of a single mutation in one or a few genes. It is a malfunctioning cell due to mutations that in the regulation of the cell cycle. So that's the part that I'm going to underline that differentiates these things. It's generally caused by mutations in genes that regulate the cell cycle. So usually it applies to a single organ. So if we think back to that process of unlocking a certain strand or region of your overall DNA, the reason that cancer so often happens to one organ in the body is because the same part is unlocked for cells in that organ. And so if those cells are cancerous, that means that the regulation of the cell cycle is damaged in that particular type of cell, okay, in that particular region of the DNA. That's why the cancer is isolated to that part. Usually, if it gets really bad, it spreads to other parts of the body as well. So big difference again, a mutation in a few genes would cause a genetic disorder, a mutation in regulation of the cell cycle that causes cancer. Other things to know is that cancer, both are very difficult depending on which genetic disorder you have, but cancer and say sickle cell anemia are treated in very different ways, as are other genetic disorders from cancer. So big takeaways, we can have neutral mutations, we can have beneficial mutations, we can have harmful mutations. It's important to know that both viruses like COVID-19 and our own body can experience genetic mutation. And it's very important to understand that the process takes place in the sequencing of DNA, all right? Sometimes it works to our favor, but sometimes it can cause great harm, and it's essential for evolution. So it is a necessary evil in a way. Outside of this particular lesson, I hope that this has been informative for you as a way to take notes and remember information. So before I was creating Google Docs for you to answer questions and review information on, now I do kind of want to get into this process of how do we take notes effectively and remember important information. This information is directly applicable to the independent study questions that are on the slides. So I'm going to leave you to do those, and I will see you next time. I hope this has been helpful for you. Take care.